Solitary bee species represent the most diverse group of bees on the planet, but it can be quite difficult to successfully rear the larvae under highly controlled conditions. We have created a method for in vitro culturing of solitary bee species, allowing us to isolate and measure risk factors associated with their microbial symbionts.
Among certain stem-nesting solitary bee species, it is critical that the culturing techniques cater to the young larvae as well as their pollen-borne microbial symbionts.
Here, we have illuminated the main and interactive effects of multiple stressors, allowing us to discern whether the stressors are largely acting alone or in concert.
Demonstrating the procedure will be Molly Bidwell and Prarthana Dharampal, both from my laboratory.
Using a sterilized scalpel, dissect freshly plugged Osmia nesting reeds. Split the reeds into 2 parts lengthwise to expose the individual chambers. Nests typically contain 8 to 14 chambers with 1 egg per chamber. Identify the male eggs using the standard method. Only male eggs are used in this experiment because of sex bias in pollen provision size. Male provisions are typically smaller than female provisions. Then, with a sterilized instrument, remove each pollen provision along with the associated egg and place them in a clean weigh boat. In the weigh boat, gently separate the egg from the provision.